What you've just seen is an oversimplified and whimsical view of protein synthesis and the ribosome. What you want to remember is that protein synthesis does not occur on isolated ribosomes. It occurs in a complicated cell, and this cell has a nucleus, which has the important function of sending instructions out into the cytoplasm to control the activities of the ribosomes, these little black dots that are present in thousands and thousands of numbers in any cell. Now, from pictures of the sort that we've just looked at, we can't tell very much about the structure of our ribosome. And so very naturally, we must begin our story about the ribosome from the point of view of the ribosome as a black box. And what this program is all about is to give this black box some form, some function, that is to say, some character. The genetic information which determines the structure of proteins is encoded in the double helical structure of DNA. The words of the genetic code are 4 in number: adenine, thymine, cytosine, and guanine. There are 2 features of this code which are relevant to our story. First, in the DNA, the sequence of nucleotides on opposing strands of the double helix are coupled to each other according to the Watson-Crick rules. Thymine, T, always pairs with adenine, A, and cytosine, C, always pairs with guanine, G, in the opposing strands. Second, it is the sequence of base pairs taken in a linear order which specifies the amino acid sequence of the corresponding proteins. The particular combinations of nucleotides which program the specific amino acids are what we mean by the genetic code. So let's see how it works. In some way, DNA unwinds, and one strand serves as a template for the transcription of messenger RNA, using essentially the same rules as for the structure of DNA, so that adenine pairs with thymine, and pairing with an adenine on the DNA, a different base called uracil in the RNA. Then pairing with cytosine, guanine, and so on. Each given sequence of 3 nucleotides specifies a particular amino acid. Such triplet sequences are called codons. The completed transcript is called the messenger, and it's destined to be translated by the ribosome into protein. Here's our Rosetta Stone, a set of 64 triplets made up of permutations of 4 ribonucleotides. As you know, there are 20 amino acids, and given the 64 triplets, there must be more than one representation for each amino acid. So, for example, with phenylalanine, we have UUU, UUC, and so on. But not all of the codons are used in the same way. So, for example, AUG, which normally in a message is instructing the binding of methional tRNA, when it appears in the beginning of a message, has a very special function. It's a punctuation mark, which tells the ribosome this is the beginning of a protein. And similarly, when we get to the end of a message, the ribosome needs instructions for stopping. And so we have 3 codons, UAA, UAG, and UGA, which have no corresponding amino acyl tRNA, but tell the ribosome stop and eject the product. Now, of course, a ribosome is not able to read a chart like this. There must be some physical basis through which the nucleotides in the codon can be translated by the ribosome into a specific amino acid. Now, for a long time, people had tried to understand this problem by trying to make the amino acid and the nucleotides fit one another in specific ways. And as was very often in the beginning of this field, It was Francis Crick who understood the following. If you take amino acids and nucleotides, there's no obvious way in which they can make a stable and highly specific complex. And Francis Crick decided that if he couldn't see a way of doing it, then nature couldn't do it. And therefore he suggested that there must be an intermediary, an adapter molecule, which can read a triplet nucleotide sequence and also bind a specific amino acid and bring them together in the right combination. Such a molecule was discovered, the so-called transfer RNA molecules. These molecules, in a very simplified form, have at one end a place for accepting a specific amino acid, at the other end a set of nucleotides which are complementary to a specific set of codons. And therefore, this adapter molecule can mediate the codon amino acid translation. What this means for our understanding of ribosome function is very simply that the ribosome does not translate amino acids, the ribosome translates transfer RNA molecules. Let's assume that the ribosome has just started translating our message, and the first amino acyl tRNA has been chosen. What does the subsequent translation process look like? The next codon to be translated is UUU, and that means that the ribosome will be looking for a phenylalanine tRNA. For instance, this one for tyrosine won't fit. Eventually, the correct one is recognized and bound by the ribosome. The site that is used to match amino acyl tRNAs with the codon is called the acceptor or A site. The tRNA, which has been holding the first amino acid, does so at the peptidyl or P site. Now that both amino acyl tRNAs are held in the right position, a peptide bond can be formed between the two amino acids. The consequence of this is that the first tRNA is detached and our growing peptide has been transferred to the tRNA in the A site. In order to continue the cycle, our peptidyl tRNA must be translocated back to the P site and the messenger RNA must be advanced one codon. Now we have completed the cycle of peptide bond formation, and you see that the peptidyl tRNA is sitting in the P site, and a new codon is in the A site. We have, in effect, returned to the same functional state at which we began the cycle. And we can imagine protein synthesis as the repetition of this cycle for each succeeding codon of the messenger. Until finally, we arrive at a codon for which there is no corresponding tRNA, for example, UAG. Such a codon corresponds to a specific signal which means stop here and release product. What we've just seen tells us something about the functions of our black box. And now it's time to concentrate our attention on the composition, the form of the black box. We want to know what is in it and how complex it is. For technical reasons, we're going to be concentrating on a bacterial ribosome. And in order to get at its ribosomes, what we're going to have to do is prepare a paste of the bacterium, homogenize it, and then we can begin our extraction. The homogenate is layered onto a sucrose gradient and centrifuged. What we recover is a fraction of ribonucleoprotein particles. These are the 70S ribosomes. If we do the same experiment at a lower magnesium concentration, we don't recover 70S ribosomes. Instead, we find two smaller particles, the 30S and 50S subunits. Such a simple experiment suggests that the 70S ribosome is made up of two unequal-sized subunits. This inference can be verified in another simple experiment. If we take only 30S particles, messenger RNA, amino acyl tRNAs, and protein factors, we find that we cannot translate a messenger. And similarly, we get the same negative result with the 50s particles. But if we mix both 30s and 50s particles along with the other ingredients of protein synthesis, the functional activity of the ribosome returns. Well, we've established that our functional 70s ribosome is made up of two subunits, the 30s and the 50s subunits. And now our natural question is, how similar or how different are these subunits from each other? If we disrupt 30s subunits, we recover a 16s RNA molecule and a protein fraction. If then we run the 30s proteins in two-dimensional electrophoresis, we can resolve after staining 21 unique proteins. We call them S1 through to S21. The 50s subunit is different. When we extract its components, we find it contains two RNA molecules, a 23s RNA molecule and a 5s RNA molecule. In addition, the proteins are different from those of the 30s subunit, and we can resolve 34 unique proteins by two-dimensional electrophoresis. These we call L1 through to L34. So, in summary, there are 3 RNA molecules and 55 proteins, each associated with a specific ribosomal subunit. And this is what we mean when we say that the ribosome is a complex entity.
We know from Chuck that this black box contains 55 protein and 3 RNA molecules. We do not know, however, what is the morphology, what is the structure of the ribosome, the 70s ribosome, nor of the 30s or the 50s subunit. We have therefore investigated ribosomes and ribosomal subunits in the electron microscope. We have looked at 70s ribosomes, and you can already discriminate here the contours of the 50s particle, which is a maple leaf or a ground-like structure, and on top of it, you see the structure of the 30s subunit. By looking now at electron micrographs of isolated subunits, we have first observed 30s subunits stained with uranyl acetate, and what we find is a structure slightly elongated and asymmetric, which resembles baby-like structure with a head, a neck, and a body from which 2 lobes emerge. In contrast, the 50s subunit has, as I said before, a maple leaf or a ground-like structure, which can be interpreted as an armchair-like structure, as you will see later. This together gives us now an idea about the gross morphology of ribosomes and ribosomal subunits. It does not, however, not add to our knowledge as to where the individual components are arranged. We have something learned about the external structure, but we do not know where the individual components, namely the various proteins and the RNA molecules, are actually located within the structure. By evaluating thousands of electron micrographs, we were able to propose a 3-dimensional model of the ribosome, which is shown here. But we know now something about the morphology, about the gross structure of the ribosome, but we know nothing as to how all these ribosomal components are arranged within the ribosome. We therefore did immunological experiments which are demonstrated to you on the 50s subunit. If we take, for example, one single protein and inject it into an animal in order to elicit antibodies against this protein, we find that a whole family of antibody population is stimulated and produced by this animal, all specific to a portion of this protein molecule. The next question, having such specific antibodies now in hands, is how are these antibodies interacting with intact 50s subunits? And what we are looking at is, in the electron microscope, is subunit dimers which are connected by a divalent antibody. So you can see on this picture there is one antibody molecule combining with its FAB arm, combining two subunits, and you can see that the antibody binds at each side to the central protuberance. Where do we find now these antibody binding sites on our model? Well, for proteins L7, L12, we found several antibody binding sites in different dimers, of course, which were found to bind to several spots around the central protuberance of the large subunit. So we can conclude that this protein must have an elongated structure in the ribosome, and from more sophisticated experiments we even know that it runs from the N-terminal to the C-terminal over here. Similar experiments have been done with other antibodies, and I'll show you an example obtained with an antibody against protein L19. Anti-L19 was found to bind at a single site in this seed region. And this single site does not necessarily mean that this protein is not elongated. In a similar manner, we have mapped the antibody binding sites of the 30S proteins. And as an example, antibodies against protein S1 binds here on the temporal region of the head, antibody to protein S21 binds here at the frontal region, whereas antibodies to protein S18 bind as well on the top, on the head of the subunit, but also on two sides far apart on the body. Hence, S18 is again an example of an elongated ribosomal protein. The three-dimensional location of the various antibody binding sites on the 50S subunit is shown in a model. So you can see that several proteins are elongated. Some occur at only one site, some like L4 on two adjacent sites. So we have elongated proteins and some proteins which could be globular. If we now look at the 30S subunit model, we see that a lot of antibody binding sites are on the head of the subunits. There are also several on the body. And again, half of the proteins have been proven to be elongated. Others are maybe globular or only ellipsoids of revolution. If we now compose these together into a 70S ribosome, we see that some of the antibody binding sites are still on the surface of the 70S intact ribosome, whereas others are now hidden and disappeared on the interface region of the two subunits.
We've heard from George how the proteins are distributed on the surfaces of the ribosomal subunits. And now the question is, what are these surfaces doing during protein synthesis? A large number of experiments involving essentially chemical tricks have been done to identify the functional domains of the ribosomal subunits. And so we can say with some confidence now that the 30S subunit is the site of all of the handling of a messenger RNA molecule. And in particular, this region of the head is most intimately involved in handling and translating a codon. This is the site also where the messenger is first placed on the ribosome and where the factors which control that orientation are bound. Similar experiments have permitted us to identify where on the 50S particle the tRNA molecule is bound and where the peptide is formed. And all of this activity takes place here in this concave plateau region that George has described for us. Now, having said that this is effectively the A site, this part of the 50S subunit, the head region of the 30S subunit, we've created a problem. On the one hand, these surfaces are very close to one another and permitted tRNA to bridge the distance between the codon under here and then the tRNA on the surface of the 50S particle. However, by the same token, we've created a problem. And the problem very simply is, how can a tRNA molecule get into this interphase region? There are two solutions to this riddle. On the one hand, it may be that we've been systematically underestimating the space between the subunits. And so there may be, in fact, a much larger tunnel which permits these macromolecular components, the tRNAs and the factors, to diffuse in and out as needed by the ribosome. The alternative solution to the problem is that we're not dealing with a static structure, but we're dealing with a structure that's moving in a coordinated way. And so that each cycle of peptide bond formation is associated with an opening followed by a closing, followed by, again, an opening permitting the tRNA to leave and a new one to enter. Well, I hope by now that we have given you some idea of the character and personality of our black box. How the movements that I've hinted at are controlled and regulated is a question that we have to leave to the future to be answered.